And what we are going to do is try to understand the differences, okay, of the different muscles. We are going to see the relationship between the nervous system and the muscular system. Okay, we already studied how the action potential reaches the axonal end. Okay, and there would occur the opening of these calcium channels and the release of the neurotransmitters. So we have to go deeper into that. Okay, what happens after that? What is the connection? How the nervous system controls the muscular system, how it works. Okay, we are going to be entering into some details of the muscle fiber to see how the contraction occurs. We are going to be studying two very important concepts that are excitation-contraction coupling, simply how we convert the electricity that comes from the neurons into mechanical energy that allows the contraction of the muscle. And then we are going to see how this contraction occurs. Okay, there is a cycle that occurs in the muscles that is called the cross bridge cycle. Okay, that explains, okay, how muscle contraction occurs. Not all of the muscle contractions. Okay, we know that this theory of the sliding filaments explains very well, for example, the concentric contractions, okay, and even the isometric contractions. But when I read about this sliding filament theory trying to explain the lengthening or eccentric contractions, I am actually not very convinced that we can explain this that way. But I don't think that we actually need to understand how it works. We simply know to understand what's going on in the patient, recognize diseases, and apply, okay, the best that we know in order to make them feel better. Now we are going to be seeing different aspects of the muscle metabolism, the role of calcium, the role of ATP, the sequence of events in the neuromuscular transmission. Okay, and also briefly, we are going to talk about how we block neuromuscular transmission. That is a more advanced knowledge, but we can mention different things that can happen when we use, for example, different drugs. Energy, we are going to classify the different fibers, muscle fibers, depending on slow fibers or fast fibers, depending on the velocity of contraction, and also their metabolism, if they are more glycolytic or more oxidative. So aerobic versus anaerobic. And what happens, why there is muscle fatigue. And also, we are going to be talking about the adaptations that the muscle has with exercise, with training. And also, we are going to introduce a concept that is going to be developed more in detail in nervous system, that is the reflexes. Okay, how the nervous system controls the muscle activity through reflexes. Okay, we don't have to be aware of everything. Okay, if let's say we are walking and we step on something, let's say a piece of glass or a nail or something, we can focus on being safe, on the pain that is that we have in the foot, but we don't have to be aware of the position of the other leg, the head, the arm, the shoulders, so we don't lose the balance. Okay, so we can focus on one important thing, but the body, the nervous system, gets in control and and creates a harmony, okay, between different other muscles, bones, ligaments, tendons that allows us to not fall. So briefly covering the functions of the muscles, in physiology, instead of saying muscle contraction, we say muscle activation. However, we are not going to enter into that terminology, okay, because that could be confusing for the student. We are going to use the regular terminology of contraction, okay, elongation, in order to understand each other. Okay, the primary action of every muscle is to contract or shorten. Even though you know that sometimes we can produce force without shortening the muscle, okay, or also lengthening, which is very important when we are gonna deposit an object on the floor. Okay, the muscle is not shortening, it's lengthening, but is still developing a force. Okay, in order to not let the object drop to the floor. Okay, so we could say either shortening, lengthening, or simply keeping a tone, constant length, and degree of contraction. These muscles are going to do their work. Okay, what are these actions? Well, movement of one bone relative to another when we are walking, when we are lifting something. Very important in speaking, the muscles of that move the jaw, the tongue itself, and movement of the eyeballs, swallowing, protection of the organs of the abdominal cavity, for example, and the muscles and the organs of the neck. Ventilation, the diaphragm, the intercostal muscles allow inspiration and expiration. Don't forget the important smooth muscle that protects, that is forming part of the bladder, the uterus, the blood vessels, keeping the tone, dilation, contraction of the blood vessels, sphincters, and the heart, of course, which is one essential muscle that unfortunately we have only one. We could have two as the kidneys. Okay, motility of the food in the gastrointestinal tract, control the blood pressure, and heat production. Okay, very important one, essential function of the muscles to produce heat to prevent that we lose the core temperature of the body. So you are aware of this way that we say that muscles contract by using the model that we call sliding filament model of the muscle contraction. Okay, we have to study these sarcomeres and this sliding model without trying to enter into details that we don't need for clinical practice. Okay, so we need to understand how it works. We need to understand the function of each of the proteins, okay, but without trying to go to tiny details that are actually not clinically important. Okay, what I mean not clinically important, I've seen many questions in exams that probably you have had in the past. Which of the following bands of the sarcomere shortens during muscle contraction, the I band, the H band? And I wonder, why do we need to know that if we are going to be clinicians? I'm not going to dedicate my life to research about muscle, and I'm not going to be looking at muscles under the electronic microscope. So I'm not going to use that knowledge in the clinical practice. Okay, so even though we like to understand that, okay, and it's beautiful when we look at that and helps us understand how muscles contract, if it's not good, not necessary for the clinical practice, better not to waste neurons in that and focus on creating strong neurons, strong connections, okay, in areas of the brain with knowledge that we actually can apply when we are working. Okay, what proteins are there in a sarcomere? The sarcomere is the contractile unit of the muscle, okay, where this sliding of the filaments occurs. And there are some proteins, okay, that are important. There are more than these, but these are the ones that we really need to know. We have the two filaments, these myofilaments of actin and myosin. The myosin is the thick one that has the myosin head, okay, that the head that binds later to actin, which is the thin filament. The thin filament has the binding sites for this myosin head. We are going to see this in more detail when we study the power stroke, when we study these cross-bridge cycles. And then we have these two proteins, tropomyosin and troponin. Okay, tropomyosin is like a filament that covers the actin. It's a filamentous protein that has attached a globular protein, round protein, that is called troponin. Remember the famous troponin of the heart, troponin C. There are specific troponins for different muscles. And then we have titin. Titin is also known as connectin, okay, which is responsible for the elasticity, the passive stretch of the muscle. Notice where they are here. Okay, we have the different filaments here. Okay, we have the thick and the thin filaments. Have the myosin here with the myosin head. You have the actin in blue. And notice that there is like a spring holding these myosin filaments, okay, and binding and holding these myosin filaments to this here, okay, that we call the Z line. Okay, so it's like a molecular spring that doesn't permit that the muscle stretch too much, and also keeps the muscle tone, the elasticity. Okay, this titin is very important, for example, and is responsible sometimes for muscle stiffness that occurs in the heart in certain cardiomyopathies. Okay, they have discovered some mutations affecting titin that make the heart very stiff. Okay, it loses the elasticity that it normally has to have to dilate and then contract, dilate and contract. Okay, so the sliding filament model says that during the contraction, we are going to see the process later, these filaments of actin and myosin slide past each other, ratchet over each other, and the sarcomere shortens. And since all the sarcomeres are placed in series, okay, in these myofibrils, the whole muscle will contract. Okay, the myofilaments don't contract or expand, they simply slide past each other, allowing the contraction of the muscle. It's like this myosin in red is pulling from these Z bands, which we have here at the ends, and the Z bands come closer to each other, separate, come closer, separate from each other. Okay, let's have a break. Okay, it's 10:52. Let's have a 10-minute break before going to the excitation-contraction coupling, so linking the neuron with the muscle. Okay, is there any question before going to the break? Let's go to this part. Okay, we are going to see what is the excitation-contraction coupling. Okay, it's a process that occurs in the muscles. Okay, that simply means the conversion of the electrical stimulus that comes from the neurons, okay, into a mechanical response that will produce movement, will produce any of the functions of the muscles. Okay, it's like the link of the signals of the nervous system, okay, expressed by the muscles who are the effectors in this case. We call that a transduction, okay, of the signals that the nervous system sends, okay, into a movement or any other muscular activity, like protection, thermogeneration, etc. So excitation has to be seen as the electrical event that occurs after the stimulation by the motor neurons, and the contraction is the physical event that occurs in this interaction between actin and myosin in this cross-bridge cycle that we are going to study next. Here we have a representation of several aspects of the excitation-contraction coupling, and also the start point of this cross-bridge cycle. Okay, we have here this neuron, okay, and the motor end plate of the muscle, okay, this neuromuscular junction. Okay, remember we have in the axon terminal the neurotransmitters in these vesicles. This could be, for example, acetylcholine. Okay, when the action potential reaches this point, calcium channels open here, and the vesicles fuse with the membrane of the neuron, releasing acetylcholine or any other neurotransmitter into the synaptic cleft. Acetylcholine will bind to receptors on the postsynaptic membrane, the motor end plate, and this binding will open sodium channels that are here in the muscle fiber, in the sarcolemma. These are ligand-gated sodium channels that will let sodium in, producing a depolarization of this region here. This depolarization is going to travel in the form of an action potential using voltage-gated sodium and potassium channels to the T-tubules that are simply invaginations of the membrane, like extensions of the membrane into the cytoplasm of the muscle cell. Okay, the action potential will travel down and is going to activate, okay, other channels that are here that are physically linked to channels that are located in the sarcoplasmic reticulum that is represented here in blue. Okay, these channels are calcium channels. These calcium channels will open, and since the calcium concentration is very high inside the sarcoplasmic reticulum compared to the cytosol, calcium will move out. Okay, the calcium concentration is going to increase in the cytosol, and now we are going to have the starting point of the cross-bridge cycle. Okay, that starts when calcium binds to troponin and we initiate the interaction between actin and myosin. Okay, that's something that we are going to see next. Before going there, let's try to see what happens with the acetylcholine at the level of the synapses. Okay, acetylcholine shouldn't stay there forever. If acetylcholine stays there, the muscle is going to continue to be excited and the contraction will not stop. And that's something we don't need. Okay, we need the muscles to produce force, to produce a power when we need it to perform an action, but not after that. Okay, so this enzyme, acetylcholinesterase, okay, is going to break down acetylcholine, okay, into two components, acetate and choline. Okay, acetate is going to diffuse. This is like vinegar, and choline is going to be reuptaken into the synaptic end, into the axon terminal, and it's going to be recycled to make more acetylcholine. Okay, we recycle that choline and we can make more neurotransmitter. Okay, we are going to be seeing, okay, in the future in physiology, and also when you study the pathophysiology of some diseases, what happens with this acetylcholinesterase, what happens in different disorders that may affect the neuromuscular junction. Okay, for example, some diseases, there are antibodies that destroy these receptors here. We can have antibodies against the calcium channels that are located here, okay, or antibodies that destroy the acetylcholinesterase. Also, there might be poisons that destroy acetylcholinesterase. Okay, so we, for example, don't inactivate acetylcholine and stays stimulating the muscle, okay, or the neurons in the case of the neurons. Now, what else is important here? Well, we can go into the type of receptors and so on, but it's better to leave that for another occasion. We don't want to create any noise talking about the types of acetylcholine receptors. That's better if we study the type of receptor in the autonomic nervous system. So these are the steps that happen, okay, during the excitation-contraction coupling. The first thing is the end plate potential that happens in this motor end plate. Okay, that is a type of local receptor potential. Then we have the action potential that moves along the membrane and into the T-tubules. Then we have the opening of the channels. Remember, there is a link between the channels in the T-tubule and the calcium channels in the sarcoplasmic reticulum. Okay, this opening produces the release of calcium. Calcium binds to troponin. We have the muscle contraction. And at the end, what happens? We have to remove the calcium from the cytoplasm and take it back into the sarcoplasmic reticulum, because if calcium continues there, the contraction is going to continue, and these cross-bridge cycles are going to repeat and repeat and repeat. And that's something that we don't need, okay, because this is a process that consumes lots of ATP, generates a lot of heat, and simply is not going to perform any action. So now let's move to the cross-bridge cycle. Okay, this is what actually produces the sliding of actin and myosin and the contraction of the muscle. Now we go to the mechanical part of this process. We were saying before that calcium goes from the sarcoplasmic reticulum into the cytosol. Then it will bind to a protein that is called troponin. Okay, every time you study this, try to make sure you remember what is troponin, what is tropomyosin. Okay, troponin is the globular protein. Okay, tropomyosin is the filamentous protein. So what happens? Calcium will bind to troponin. Troponin is attached to tropomyosin, forming a complex. So this complex will move away, okay, when calcium binds to it. This moving away will expose the binding sites, the active binding sites that are on actin, okay, that will be used by myosin, this myosin head, in order to produce the contraction. Okay, so there are several steps, 4 steps in this cross-bridge cycle. The step 1 is the formation of the cross-bridge, when the myosin head binds to actin, to this active binding site, okay, forming the cross-bridge. Now, before that happens, because remember this is a cycle, okay, so something has to happen before. Before that cross-bridge formation, the myosin head, this like hammer, has to be activated. Now, activation of this occurs when the ATP molecule that was present from the previous contraction, from the previous cycle, is hydrolyzed. Okay, ATP is hydrolyzed into ADP, meaning 2, and phosphate, inorganic phosphate. Now, the energy from that chemical reaction, the energy from the hydrolysis will be kept, okay, in the myosin head, okay, after the phosphate is released. Okay, so let's try to be in the situation, okay, the myosin head is activated from the previous contraction, has a lot of energy, holding the energy, potential energy. Now the step 2 is the power stroke. It's the moment when the myosin head pulls, okay, from actin and brings the 2 Z lines together. Okay, that power stroke. Okay, what happens there? Myosin with the ADP that is present from the previous contraction pivots towards the center of the sarcomere, dragging actin, and at the end of this, ADP is released. Now, we have to continue, okay? We can leave the muscle contracted. We have to release the sarcomere now, okay? The step 3 is the cross-bridge detachment. For that, we need another ATP that binds there. When ATP binds there, the myosin head, the bond between the myosin head and actin is weakened. Okay, so the myosin head lets go of actin. Myosin detaches from actin. Now, we have to produce another contraction. The step 4 is the reactivation of the myosin head. Okay, the ATP that bound to weaken the bond now is hydrolyzed in ADP and inorganic phosphate. This reactivates the myosin head that will return to the cocked position, which is a high energy position with lots of potential energy. And these cross bridges, these cycles will repeat, okay, while the actin sites are exposed because of the presence of calcium in the cytoplasm. Okay, after the nerve stops stimulating, calcium is going to be taken back into the sarcoplasmic reticulum and the muscle contraction will stop. Okay, there is an animation. Okay, so you can see this in motion. Okay, it's very difficult. I know that is very difficult to understand how this happens just by reading or by listening to someone, because this is something that is very abstract. Okay, so if we see this in motion, okay, it's going to be a lot better for us. Okay, I put there in the notes, when you look at the notes of the PowerPoint, I put there a series of links with videos from YouTube that helps us to have this very clear. So just to remind a little bit about this sarcomere, okay, there are different components. The Z line, a sarcomere goes from Z to Z. Okay, we have these thin filaments of actin, we have the thick filaments of myosin with the little heads there, the hammers or heads, okay, that are bound in the center by another protein or some proteins that form this line, the AM line. Okay, when the contraction occurs, simply these Z lines are going to come closer, okay, will be dragged toward the M line, okay, and all of these sarcomeres that are placed, organized in series, are going to do the same. Everything is happening at the same time, so all the muscle will contract as one. And we have this beautiful thing here in this electronic microphotograph. Okay, so we can see how it looks like, okay, what we call the M line, what we call the Z lines with the different proteins there that bind the actin to the Z line, the myosin to the M line. Okay, we can see the myosin heads there. They don't represent titin here, but titin should be here, this molecular spring. Then we have lots of things around that you can see. All of these things are mitochondria, are granules that contain glycogen, etc., that allow the functioning of these muscles. Okay, when you study the histology, you learn a lot, okay, about how things work and why different organs, different tissues are organized in a different way. And you see how all of the organs are like little factories. Factories, for example, if you make ice cream, you have lots of sugar and milk. But if you make shoes, you will have leather, you will have different other things. The same happens in our body. And the more you study these details, the more you get amazed about the organization of the tissues and our body. Now, mitochondria, glycogen. These are things that work in the production of energy for the muscles. Okay, glycogen is a form of storage of energy. Mitochondria take different materials like pyruvate, like fatty acids to make ATP. Okay, when the muscle needs to contract, the glycogen is broken down to produce glucose that can be used to make ATP or can be used to make pyruvate, and the pyruvate taken to the mitochondria for energy production. Okay, and all of these will depend on what type of exercise we are doing. So let's take a look at the most important metabolic systems that exist in the muscle. Okay, there are different metabolic systems, and all of them are going to produce something called ATP, which is extremely important for different processes in the muscle. Remember, just to maintain the resting membrane potential, we need ATP, because ATP is necessary to maintain the concentration of potassium inside the cells, different from the concentration of potassium and sodium outside the cells. Then we are going to need ATP for this cross-bridge cycle, for the detachment of the actin and myosin, okay, and also to produce the power stroke, the hydrolysis of this ATP. Then we need ATP to take the calcium back into the sarcoplasmic reticulum. Okay, different things ATP is doing there besides participating in other processes that normally cells do. Okay, so ATP, for example, when it detaches actin from myosin, it helps overcome rigor. Okay, there is when people die, okay, the muscles at the beginning are very rigid. We call this rigor mortis, And this occurs simply because of the lack of ATP. Okay, when the cells die and there is no oxygen, ATP is not made. So these actin and myosin are gonna be very tightly bound to each other. So it's impossible to relax the muscles. Okay, then when the proteins of the muscles start breaking down as a result of the normal process of necrosis, of course they are gonna be lax, but this is simply because of the breaking down of the proteins. Okay, so many different functions for ATP there. And basically we have some ways of obtaining ATP that we can summarize with these three. Okay, we have what we call the phosphagen system. That is a system that provides energy for a very, very tiny time. Okay, when these mitochondria produce ATP, okay, some of it will be stored as creatine phosphate. Okay, this creatine molecule is gonna keep one phosphate. So every time the muscle contracts and an ADP is released, it will bind the phosphate to the ADP to form ATP. But we have very little of this. Provides energy for just 10 to 15 seconds. So maybe if you're in a place and you see something and you have to jump, or maybe for that is good. For that, we also can use the little ATP that is available in the cytoplasm. Then we have the glycolytic system, or also known as glycogen lactic acid system. It's something that we know as anaerobic respiration. Okay, that it provides little ATP, just 2 ATP per every molecule of glucose. Okay, that system provides energy for, let's say, 30, 40 seconds, and has something that we don't like, that it creates lactate. Okay, lactate is something that reduces the pH inside the cells, and this acidosis will impair some of the functions of the muscles, leading, for example, to muscle fatigue. And also, if we produce lots of lactic acid, this lactic acid may go into the circulation, producing acidosis in the body, and we don't like that. Okay, changing the pH of the blood may impair some of the functions of the hormones when they have to bind to different receptors, different chemical reactions. If the pH of the blood is very low, are not going to occur very well. So lactate is something that we have to eliminate, okay, as soon as we can. And then we have the aerobic system, also known as oxidative phosphorylation, is the one that occurs in the mitochondria. That is the best one. Okay, for example, it's the only one that our heart prefers. The heart doesn't like working under anaerobic conditions. Okay, it doesn't have many means to do that, and it's not resistant to that. Okay, for example, every time we do aerobic exercise, if we are jogging, for example, or running, jogging on a treadmill, we can stay, depending on the training that we have, we can be there 1 hour, 1 hour and a half, or more. Okay, think on people who run marathons. How many hours are they running? Okay, of course they are tired, they have pains in the joints. Okay, they are dehydrated, they are feeling bad sometimes, but they are not making lots of lactate. If they were making lactate, they would have to stop. Okay, they are using this aerobic metabolism, oxidative phosphorylation, that produces ATP, 30, 32 ATPs, depending on the training, depending on the integrity of the mitochondria, and produces CO2, carbon dioxide, that we can easily eliminate just by breathing out, and water that we're going to sweat or we are going to urinate. Okay, the aerobic system can use glucose, can use fatty acids, and can use some amino acids as well. And for example, the heart, okay, uses fatty acids, okay, in order to perform its metabolism and keep going on during 80, 90, 100 years with no stop. Now, what happens with the aerobic system is slow. Okay, so if we are doing a sprint, we're running very fast or we are lifting weights or something, it's going to be very hard to use it, okay, because it requires lots of oxygen and it's a slow system. We have to take pyruvate from the glycolytic pathway and take it into the mitochondria, okay, and produce a series of steps in order to make these 32 ATPs. Okay, so during aerobic exercise, this is great, but doesn't work if we are doing fast exercises, different type of sprints, etc. This diagram is great to understand this. Okay, here we have the creatine phosphate system. Okay, notice that here we have ATP. Okay, ATP will bind to creatine, okay, forming creatine phosphate and adenosine diphosphate. Okay, creatine phosphate, by the action of the enzyme creatine kinase, will take this ADP and add a phosphate to it, producing another ATP. So every time we use an ATP for a muscle contraction, the ADP can be reconverted to ATP. It's like a recycling, okay? So we have a constant supply of ATP in the muscles. Then we have here the glycolytic pathway. Okay, notice that either using glucose from the blood or glucose from the breakdown of glycogen, okay, this glucose may be broken down, glycolysis, to produce a couple of ATPs and also a couple of molecules of pyruvate. Okay, in the case that we are doing aerobic exercise and there is oxygen, this pyruvate is going to be taken to the mitochondria to make lots of more ATP. But if there is no oxygen, the pyruvate is going to be converted into lactic acid, and that's the problem here. Okay, making the cytosol of the muscle or the blood too acidic. And that's, this one down here is the best. Okay, cellular respiration in the mitochondria, aerobic respiration in the presence of oxygen, glucose, pyruvic acid, fatty acids, okay, will be used in the mitochondria to produce tons of ATP, 30 to 32 molecules, and carbon dioxide and water that we can easily eliminate. Carbon dioxide is not going to produce acidosis unless someone has a respiratory problem. Because let's say someone has pulmonary edema. The CO2 is going to travel in the circulation, and when it reaches the lungs, it cannot be eliminated because there is water in the lungs. So in that case, it may produce acidosis, but not normally. And also produces heat, ATP, water that can be eliminated. So there is no danger of wastes accumulating in the case of we use this aerobic metabolism. Okay, and something that I'm sure you know very well is for how long we can use the different systems. Okay, first seconds we use the ATP that is stored, freely floating in the cytosol of the muscles. Okay, then we use the one that is provided by the creatine phosphate system for 10, 15 seconds. Then we can use the glycogen, okay, to that is stored, or glucose from the blood in the anaerobic pathway, 30, 40 seconds. But remember, if we are doing a very fast sprinting, that's going to represent a quick end of the exercise because of the accumulation of lactic acid. Muscle fatigue, acidosis, and the last one that can be for hours, okay, is the aerobic respiration. Okay, theoretically, if the exercise depended only on the muscles, that could be there. We could be exercising forever. However, we don't, because remember, the joints are suffering, we are getting dehydrated, our blood pressure changes, our potassium, sodium, etc. is altered. Okay, all the body is suffering from this excessive exercise, and at the end, we have to stop. Okay, and this diagram here represents more or less the same, but in a graph. Okay, the immediate system, creatine phosphate, for just some seconds, glycolytic pathway, maybe 30, 45 minutes, and then long-term mitochondrial respiration that theoretically could be extended forever. Of course, we know that that is not true, but that's simply theory. If we isolate the muscle from the body, it might be working forever. Now, the metabolism of the muscle fiber is useful to understand the classification of the skeletal muscle fibers. Okay, we can classify the skeletal muscle fibers using a couple of things. First of all, the velocity of shortening, how fast it contracts. Okay, the contraction velocity depends on the activity of an enzyme that is present in the myosin head, okay, that can act more quickly or more slowly. Okay, this enzyme is called myosin ATPase. Okay, some muscle fibers have a very fast ATPase, some others have a slow ATPase. So muscles can shorten either fast or slowly. Okay, the fast fibers will be more prone to fatigue. If they are contracting very fast, of course, they are gonna fatigue earlier than the slow fibers. If they are working more slowly, okay, they are be less prone to fatigue. Slow fibers are the ones that are capable of maintaining smooth tetanic contractions for a long period. And I think it's important to make a stop here and explain these tetanic contractions, because some students in the past have asked me, but tetanic, that's not a disease, it's not bad. Well, not exactly. The muscle contractions can be classified in different ways. For example, a twitch. A twitch is simply, let's say you have a stimulus, an action potential reaches the muscle fiber, and there is going to be a contraction and a relaxation. That is a muscle twitch, contraction, relaxation. Or you can produce several stimuli. Let me represent here a graph with the frequency of the stimuli. Okay, if we produce the stimuli very far away in time, very apart in time, we are going to have a twitch, another twitch, another twitch, another twitch. Okay, contraction and relaxation. But if we put the stimuli more closer to each other, we are going to have a twitch, and when the muscle is starting to relax, there is another twitch. And when it starts to relax, there is another twitch. Starts relaxing, another twitch. That is called summation. Exactly as the type of summation that we studied in the neuron. Okay? We add the effects of the different stimuli, so we obtain a larger one. But notice that the next contraction occurs before the first twitch actually ended. Or we can have, in the case of having the stimuli closer, very close to each other, we will obtain a straight line. So no relaxation at all. A sustained contraction is what we call a tetanic contraction. Tetanic contraction. For example, if I am watching TV, the muscles of my neck are in a sustained contraction. They are not relaxing at all. And I need that in order to keep the head in position. When you're holding an object, we need a tetanic contraction of the muscles. I don't need to be shaking. Okay. So tetanic in this case is not something bad. Now there are diseases. For example, after infection with Clostridium tetani, there is a toxin that produces tetanus, but that is pathologic. Okay, so the velocity of shortening is one of the things that we use for classification, and then the major metabolic pathway, if it's either oxidative or glycolytic. Okay, there are different fibers. Okay, some time ago, the classification was type 1, type 2A, or type 2B. Now they have changed this a little bit because it is very hard to remember. Okay, which is the type 1, 2A, 2B. That doesn't say anything. Okay, mind that we call our kids number 1, number 2, number 3. Okay, if they are twins, 2A and 2B. No, that's not a good idea. Okay, no one will remember our names ever. Okay, so the previous type 1 fibers, that was good because I remember when I studied medicine, this was the classification. And I used to say, okay, the type 1 are the first quality meat. First quality. It's very expensive, like filet mignon. Okay, so I could remember by the color of the skin, how they were inside. Okay, what was their function, paravertebral muscles, etc. Okay, these fibers, the ones that we use, for example, in the paravertebral muscles to sustain our posture, the posture of the head, the posture of the back. Okay, these fibers contain slow oxidative fibers. These are muscles that are constantly working. We are the whole day holding our head in some position on our back, either sitting or standing or walking. We have to maintain the stability of the trunk. We don't do very fast contractions, at least normally. Okay, we are not constantly moving our head in different positions. So these fibers are slow, produce slow contractions, and are oxidative. They have a slow activity of the myosin ATPase and very high oxidative capacity. Constant ATP production by the mitochondria. That means they need oxygen, lots of oxygen, and they need a lot of mitochondria. Now, these fibers shouldn't rely on the oxygen from the blood because they need to be working all the time, even if the oxygen supply is reduced. So they have a way of storing the oxygen inside. They have a molecule that is called myoglobin that is very similar to hemoglobin. Okay, hemoglobin is the molecule that we use to transport oxygen throughout the tissues, throughout the body, in the red blood cells. Well, myoglobin is the same, but in the muscles. Okay, when blood moves through, the muscles get the oxygen, use part of it, and the other will be bound to myoglobin. Okay, so when we are in any situation in which we are exercising a little bit and there is not very good supply of oxygen, we can use the oxygen from myoglobin and the activity of the muscles don't stop. And it's because of the presence of myoglobin that these muscles are very red. Thinking filet mignon, thinking the color of this meat, and compare it to other types of meat that is more whitish, less red. Then we have the other types of fibers, the type 2, divided in A and B, because the A is fast contraction but uses oxidative metabolism, while the B has fast contractions but uses glycolytic metabolism. The fast contractions are because of the very high speed of the myosin ATPase, and the oxidative is exactly as the one above. They use lots of mitochondria, not as many as the type 1. And the fast glycolytic, or 2B, are very similar to the type 2A, but instead of mitochondria, they use mostly glycolysis, glycogen. So they are not going to have too much myoglobin. They don't have too many mitochondria. They have lots of glycogen, which is yellowish or whitish, white yellow. So think in the color of the meat again, red, beautiful bright red, or pale yellowish, pinkish, anemic meat. And think in the price. First quality meat is very expensive. You go to a restaurant, don't even ask for this. You have to ask for a loan. But if you eat the regular mixture of fibers, it's not going to be too expensive. The type 1 predominating the postural muscles, very resistant to fatigue, while the type 2 are used for rapid movements. They fatigue more easily. Now, these fibers are in combinations, okay? For example, the paravertebral muscles will have lots of type 1, while the muscles in the legs are gonna have more of these type 2 fibers, because that's the ones that we use for sprinting, for jumping, for doing very fast movements that maybe the other ones can do. For those who like tables, okay, there is one here, okay, that you might like. We are comparing here the type 2 and the type 1. Okay, we're not making here the too much division between the 2B and 2A and 2A. We're gonna see these as two groups simply: fast twitch and slow twitch. Fast, high velocity; slow, slow velocity. Fatigue: if you contract too fast, it is very easy that you get fatigued. If you go slowly, you're gonna be more resistant. Now, why they are able to produce these very fast twitches? Remember I told you that mitochondria produce ATP very slowly. Okay, these fibers have more glycolytic metabolism, more glycogen. That's why what explains their color, okay, and what explains the content of these fibers. Very low glycogen in the type 1. That's why they are red, because they have more myoglobin. Oxidative, these fast twitches don't produce too much oxidative metabolism. They don't need too many mitochondria, and they don't need myoglobin because they don't need to store glycogen if they are not going to use it. In the case of the type 1, all of these factors are high: myoglobin, mitochondria, and oxidative capacity. And what is the type of, in case of sports, what is the type of people who have a higher proportion of the different fibers? Well, long distance runners will have a higher amount of type 1. Middle distance runners will have a 50-50. And sprinters will have more fibers 2 than 1. And the example that I like better, this one. I think this is something that everyone understands. Notice this piece of meat. There is a reddish portion. At least when I'm going to the supermarket, I'm looking for this, the tray that has more of this reddish thing, because that is very soft. Why soft? Because it has mitochondria. Mitochondria are fat. That's why it's very soft. This white part doesn't have mitochondria. Okay, it has more collagen and more connective tissue. That part is not very good. Okay, red muscles have a lot more myoglobin, has more iron, has more myoglobin. Okay, that's why it looks red. And even you're gonna learn, if you go to this link here, you're gonna learn many interesting things about the color of the meat in the supermarket. For example, when the meat is stored under a vacuum, it is in a reduced state and becomes purple red. If exposed to oxygen, Myoglobin is oxidized to oxyhemoglobin, which has a very attractive color. During storage, it's converted to myoglobin, which is brown and unattractive. That's why they put some gases in the trays to make it keep the bright red color, and we like it and we buy it. Okay, the color of the meat also depends on the diet of the cow. If you feed the calves with cow milk that has no iron, it's going to have very little myoglobin. That's why the calf's meat, okay, is going to be pale compared to a cow's milk. Okay, if you read that, you're going to learn very interesting things. I love this thing, and I love learning about this stuff. So the last thing that we are going to be talking today about is muscle fatigue. Okay, the rest is going to be for the next encounter on Wednesday. And I think that at this point, we already have mentioned many of the factors that contribute to muscle fatigue. Okay, the most important one is this accumulation of lactic acid that lowers the pH inside the cells, leading to impairment of different functions. Fatigue has different meanings, okay? When we say we have fatigue, we are talking about tiredness, lack of energy, exhaustion. We can have mental or physical fatigue. Now, muscle fatigue, what it means is a decrease in the maximal force or power. There is a difference between force and power. Force in a muscle equals strength. It's the capacity that we have to move something or hold something or change something. Now, what is power? Power is force times velocity. So power is the capacity to do something rapidly. How quickly we can do something. Because maybe I can hold this cup of water and I can lift, but I can lift it very fast or very strongly. So I have the force, the strength to lift the cup, but it will take me forever because my muscles are very weak. So if I am able to do this very fast, I have a lot of power. If I have to do it very slowly, I have little power, even though I have force. Okay, so it's important to understand concepts, because when we say power, force, we are not saying the same, at least in physiology. Okay, when you talk with other people that don't study physiology, it's the same. I have a lot of power, I have a lot of force, I have a lot of strength. It's the same in this talking colloquially, but in science, every word has a meaning, which is important to remember. Okay, so accumulation of lactic acid, phosphate. Reactive oxygen species is going to impair all the functions. For example, low pH doesn't let the sarcoplasmic reticulum to release calcium and reduces the sensitivity of troponin to calcium, so the cross bridge is not going to be made properly. Okay, we are going to stop here. And there you have more for reading. There is an article with potential solutions for muscle fatigue. I know some of you will like it if you are in training, exercising, in physiology of exercise. You are going to enjoy it. Okay, I hope you learned something, and I hope that I can learn also from you. Okay, so please write me, tell me what you think, what ideas you have, what could we incorporate if we wanted me to talk about something, okay, or stop something about something. Okay, any